Hello, my name is Hannah Kim. I am a fourth-year bioinformatics PhD student from Temple University. Today, I would like to talk about my project, Prime Evolutionary Imputation. First, our motivation. Let's start with an example. What you see here is the trimer of SARS-CoV-2 spike protein binding an antibody. The infamous M501Y mutation found in most variants of concerns is right here. A radical amino acid change like this indicates changes in function, and amino acids in different parts of the protein will be under different physicochemical constraints. However, most previous evolutionary models treat amino acid altering changes as equally likely and average them out across the entire gene. Even the conventional amino acid exchangeability matrices like PAM and BLOSSOM do not take physicochemical properties into account. There, we asked ourselves, can we identify properties that are under selective pressure at each site? Furthermore, can we define sample space in which a site can evolve? These were the ideas behind PRIME. PRIME is an abbreviation for Property-Informed Models of Evolution. PRIME takes a multiple sequence alignment and a phylogenetic tree as inputs and returns properties that are changing or conserved. Note that PRIME has site-level resolution. Briefly, our method can be summarized in 3 steps. First, PRIME builds on top of evolutionary inference framework that takes amino acid site specificity into account. Second, it estimates physicochemical property distances of the input alignment. Vectors of predefined property values for each amino acid is used. Take glutamine and lysine, for instance. Each tip on the radar plot represents a value from each physicochemical property. The difference between the same tip of the two plots, that's what we define as the property distance. The property tree in this case is volume, and this is how we define volume distance between glutamine and lysine at a particular site. Lastly, using the distances from step 2, PRIME performs maximum likelihood estimation for property parameters which lead to the output mentioned in the beginning. PREI is an abbreviation for Prime Evolutionary Imputation. The imputation process is as follows. Given the topology of the input tree, we are interested in site 8 of sample X. We mask this node and use the rest of the nodes and the prime estimated property parameters to impute the probability of codon character states, which we call evolutionary credibility. Note that currently at site 8 of sample X, we have GAT, which is translated into aspartate. Most PREI results will return the highest evolutionary credibility with the same amino acid as expected. But here, we present to you an interesting result where the amino acid with top evolutionary credibility is not the same as the current amino acid. GAT is ranked 5th based on our imputation, and it is one of many codons that can occur at site 8 based on evolutionary inference and physicochemical properties. PREI has identified the sample space of change in site-specific and sample-specific manner, and we know what it may evolve into next. PRIME and PREI have many applications. Our methods can detect sites going through functionally important changes, and they can help forecast short-range evolution of one species from the long evolutionary history across species. Particularly in the context of infectious diseases, that means this information can be used to find targets for drugs and vaccines and be used to monitor natural virus reservoir and to project important changes. Our methods are not restricted to viruses, so it can be used to study chloroplastin, for example, to understand important functional changes and physicochemical properties that affect it. I would like to acknowledge my advisor, Dr. Sergey Pond, and the members of the ACME group for their help and encouragement. I would like to use this opportunity to make a plug for the newly formed Center for Viral Evolution, chaired by Dr. Pond. We hope to do a lot of exciting research on viruses. Today, in the interest of time, I was only able to provide you with summaries of our methods. For more information, please follow these links for code and visualization. Remember the handle, Hannah Kim Campayo and S Pond. Thank you all for listening.